You guys, my hand comes down, Jeff. Look at right.
Well, welcome to ASM Live for 2011. We're here in New Orleans at the ASM General Meeting. I'm Jeffrey Fox of Microbe, the monthly magazine of ASM. And here in our first session, I will be talking with Traci Testerman of Louisiana State University in Shreveport. And Traci is going to be talking to us about her work on Helicobacter pylori and finding a connection with Parkinson's disease in a mouse model that is studies in mice. And I'm going to ask Traci to give us a brief summary of some of the highlights of your work, and then we'll get into questions and answers. Before you talk, I should remind those of you in the webcast audience that you can ask questions by either through the chat room or hashtag ASM 2011. And for any of you here in our live audience in the room, if you'd like to ask a question, please raise your hand. Someone will bring you a microphone and we'll ask you to name yourself and your institution, ask your question, and we're eager to have them. But first, Traci, tell us about your work.
Okay, well, I have been studying Helicobacter pylori for a number of years now, and Helicobacter is an organism that was not discovered until 1984, and it colonizes the stomach of about half the world's population, so it's extremely common. And it causes ulcers in about 10% of the people who are infected. And it can also cause gastric cancer in a small percentage. But most people who are infected don't even know that they're infected. It doesn't necessarily cause any symptoms, or it may be just some mild dyspepsia type symptoms. So currently, Helicobacter infection is not treated unless there are some kind of problems like an ulcer. And so before I started the Parkinson's research, I was studying adherence of Helicobacter to host tissues. And I had found that two proteins called ALP-A and ALP-B bind laminin, which is a member of the extracellular matrix. And so I was interested in what effect the loss of laminin binding would have on pathogenesis. So I expected that this mutant lacking both of these proteins would not colonize as well as the wild type because it wouldn't be able to stick as well in the stomach. And in mice, it doesn't colonize quite as well. However, we noticed in gerbils, the gerbil model, they have pathogenesis very similar to humans, where they can get ulcers. And so in that model, I noticed that this mutant caused more inflammation than the wild type strain, which was really a surprise. So I just happened to have some animals that were infected with the wild type and the ALP-A/B mutant. And an emeritus member of the faculty had brought to our attention a hypothesis paper hypothesizing that a modified cholesterol made by Helicobacter is neurotoxic. And so I'm a microbiologist, and I teamed up with Dr. Michael Salvatore, who's a neuroscience researcher and studies Parkinson's disease. And we decided one day just, well, let's take a look at the brains of these animals that I had infected. And so there were only a few of them, not enough to get statistics, but he took a look and he found that dopamine levels were reduced, and he thought, let's take a closer look at that. That looks interesting. And he noticed that it was more dominant. The loss of dopamine was greater in the ALP-A/B mutant than in the wild type. So he wanted to try those same strains again.
And I'm a little confused here. Were the animals had no overt symptoms of a
No.
neurologic disorder? You were just teaming up with a neuroscientist and
Right.
looking at blood levels or something of a particular neurotransmitter?
The dopamine was measured in certain regions of the brain. The regions that we've
okay.
been looking at are called the dorsal striatum and substantia nigra, and these regions are very deep inside the brain, and there's degeneration of the neurons in these regions during Parkinson's disease in both humans and in animal models.
Okay.
So he took the brains out and removed certain regions, and he tested for dopamine, and he found that the levels were decreased. So we then did a larger experiment, and as we all know, older people are more likely to have Parkinson's disease than younger people. It's usually diagnosed after the age 50. So we wanted to look at young mice and old mice. So we got some mice that were the equivalent of late middle age, or middle age to late middle age, and also some younger mice, some teenage ones. And we infected them with these strains. And we measured over time the locomotor parameters. So what we do for that is we have a chamber. You just put the mouse in it, let the mouse run around, and there are sensors to detect how much the mouse moves and what types of movements. So it can track speed, it can track distance, it can track starts and stops. So this allows us to see how well the mouse is moving. And we saw declines over time in the infected mice in the old group, but not in the young group.
Okay. And these mice were, what kind of period in terms of the equivalent to human years was this over weeks, months, years of, no, not years with mice, but.
Right, well, obviously mice age a little faster than humans do. So we were going from, age of maybe 50, 60 up to maybe an age of 80 or so, which is a few months, in about 5 months.
Okay.
In mouse time. So this would be the equivalent of an infection in humans that would last over a number of years.
All right, so you're really modeling, tracking what happens in Parkinson's disease in humans with this kind of time span in mice. That's
right.
The point. And so what's your idea of how is Helicobacter, which is after all in the gastric upper GI tract or in the stomach, how is it affecting the brain? What's your idea of how this happens?
Well, the main hypothesis is that a modified cholesterol produced by Helicobacter is being taken up and eventually trafficked to the brain. So what Helicobacter does is it steals cholesterol from the host. It can't make its own cholesterol. So it steals it and it adds a sugar group to it, and then it puts that in its membrane. And the chemical structure of this is very, very similar to the structure of a known neurotoxin produced by a cycad plant.
Okay.
So that was where the original hypothesis came from. That was put forth by Christopher Shaw. And so that definitely is the leading hypothesis. And what we've actually shown is that you can cause the same symptoms if you feed killed Helicobacter to the mice over a period of months. So you can get it by feeding or by infection. Now, one reason we think that...
I do have to ask.
Okay.
Does Helicobacter lower cholesterol levels in the mice?
No,
Doesn't make them healthier?
No, it doesn't.
No, there's no plus there.
In fact, Helicobacter is believed to contribute to atherosclerosis, so it's not
pity.
helpful in that regard.
All right.
Yeah.
Now, you haven't tried, I'll ask a more serious question. Is it possible to use purified cholesterol, this sugar-modified cholesterol, to do similar experiments and reproduce the symptoms, or do you have to do killed Helicobacter, which makes me think that maybe there's more in the mix.
Right. Yes, we haven't used the purified compound yet. However, the researchers that are studying the cycad toxin, they have purified this substance, and they've either injected it into the brain or given it orally, and they've found that they get symptoms produced. So we have reason to believe that it will work, even though we haven't tried it yet.
Okay, so you still don't, so there might be other cofactors.
Right. Well,
in
and we believe that there are, because the strains of Helicobacter don't behave the same. And we don't think that they have different levels of the modified cholesterol in them. We think that other factors, such as perhaps inflammation, could also be contributing to the disease.
No, and I wanted to go back to another point you had made, which was that you talked about, I believe the mutant does not adhere as well to the surface in the mouse stomach. Why would that make a difference in terms of the outcome of your findings? Do you understand what's going on there?
Well, we haven't figured out exactly why that mutant strain does cause more inflammation, but what I believe could be happening is that Helicobacter is actually interacting with the host cells to try to reduce the inflammation, because too much inflammation is actually bad for the Helicobacter. They don't like that. They tend to get killed. So I believe that they may actually be acting to reduce this, and by not having contact or having less contact with the host cells, they're not able to inhibit the inflammation.
Okay. Now, another question comes to mind too, which is you've used young and old mice, but I'm assuming that these are young and old mice of the same type.
Right.
So would you expect any differences in terms of if you used a different strain of mice or host factors that might make a difference in terms of the outcome, the tendency to develop a Parkinson's-like syndrome?
Yeah, so far we've only looked in the one mouse strain, but it is widely known that different mouse strains differ in their responses to infection and to other things. So I think it's quite possible that different mouse strains would behave a little differently. Maybe some would be less susceptible or more susceptible. And
okay. And the gerbils, have you looked at them further, or?
We haven't looked at gerbils yet. One of the reasons is that the assays in the brain use certain antibodies, and those are for mice, and we don't know if they'll cross-react. But we would like to try it in the gerbil model, because I think that could very well work as well.
And do you have any, of course we have enough trouble dealing with cholesterol as an enemy of good health, but I suppose you could come up with something that would target that sugar as a potential treatment to counteract what's happening?
Yeah, that's what we're hoping, is that we will be able to come up with some specific treatments that target that, because that cholesterol is very important for the Helicobacter, and it's also important that it have the sugar group on it. We have a mutant strain developed by a collaborator of mine, David McGee, who also studies Helicobacter, and it's a mutant that is unable to put the sugar group on. And so those bacteria do not infect very well. So yes, a specific drug would be very useful. And it would also be useful just in general for treating Helicobacter, because antibiotic resistance is becoming an increasing problem. So it's getting harder and harder to cure Helicobacter infections.
Oh, is that right? But typically a sort of heavy-duty dose of several antibiotics at once is?
Yeah, typically there are 2 antibiotics given, and then a proton pump inhibitor, similar to what people take if they have bad heartburn. Typically that combination, given for about a week, that usually works about 90% of the time. But as Helicobacter becomes resistant to more and more antibiotics, it gets harder. So there are some known strains that are resistant to all of the commonly used antibiotics. So that really poses a problem for treatment.
Okay, and of course you're talking here of something that would be a chronic insult. So if you could get in early, I mean, if this all holds up, of course. I think it would be interesting to explore this idea a bit. I don't have a good question in mind, but.
So if you treat the Helicobacter early, you could maybe prevent some of.
Well, or at least maybe you want to keep leave the Helicobacter alone, but have something that would be a benign treatment that would somehow block any possibility of developing Parkinson's symptoms, if that were possible.
Yes, we're planning to look into that as well, because, and there's an urgent reason for that, and that is there's a group in the UK, John and Sylvia Dobbs, and also there's another one, blanking on the name right now, but they have done a clinical study, actually, where they took Parkinson's disease patients who were infected with Helicobacter, and initially they either gave them placebo or active treatment, and then they followed them. And what they saw was that the patients who were successfully cured of their Helicobacter actually improved a little bit in their ability to move. However, the patients who were given the antibiotics but didn't clear the infection, they got dramatically worse. And so later they offered all of the placebos antibiotics, and they found that they then improved a little bit, or at least didn't progress as much. But there is that caveat that, I mean, they had, I think they had about 20% eradication failure in their study. So that's a big risk. If you wanted to treat a Parkinson's disease patient with antibiotics, that's a big risk, and we would like to at least find a way that you could prevent that downside from happening if the antibiotics don't work. So then you could just try again with different antibiotics and not cause the patient any harm.
No, I wanted to, it occurred to me that it would be an interesting experiment, even if you can't get the mutant of Helicobacter that can't put the sugar on the cholesterol to colonize the mice. You can do a killed Helicobacter mutant study to see whether you get the Parkinson's symptoms.
We're actually just starting that experiment right now. So
it sounds like a good experiment.
Yeah, maybe 5 or 6 months from now we'll have an answer on that. But yes, we're just getting started on that.
All right. We have a live audience here, none of whom want to ask questions, and it doesn't seem as if we've got questions from the. Oh, we do have questions. Good.
Hi, I'll start. I'm Michael Schmidt. I'm on the faculty at the Medical University of South Carolina. This is an absolutely fascinating story, going back to one of Dr. Schechter's small things considered questions. Have you ever identified a disease for which there's not a microbial trigger? And I think now with Parkinson's, we again have that wonderful microbial trigger with Helicobacter. One of the interesting things is Helicobacter is vertically transmitted. You get it from your dear darling first caregiver, your mother, your father, your grandparents. And so this is an organism that comes with us from the beginning of time, as it were. So I guess the real question is, when should we think about going after Helicobacter? When you're young, before the cholesterol has invaded our brain and killed our dopamine producers? Or do we wait until we're in our 50s, 60s before we develop the symptoms? When do you go after that treatment? And then the second question is, can we make a vaccine to Helicobacter that's living in that protected niche of our wonderful mucosal layer in our stomach, where it's effectively hiding from all things immunological? Thank you.
Okay. Well, first of all, there are a number of groups working on vaccines, working very hard, but I don't think that we're going to be seeing an effective vaccine, at least within the next 5 years. I don't know about further out. So, yeah, that would be great if you could come up with a vaccine. So as far as when to treat Helicobacter, now there are some people that think that Helicobacter may be useful during childhood in preventing allergies, but it's not clear whether the Helicobacter are actually responsible for that, or whether something that goes with Helicobacter infection, another organism perhaps, is responsible. So it's really not clear whether it needs to be treated in childhood, but I think definitely by early adulthood we should be treating for it. And hopefully that would lead to prevention of a number of cases of Parkinson's disease and maybe some atherosclerosis, gastric cancer, these kinds of things, because these things all seem to develop over a very long time period of infection.
Okay, we have a question from online, from the Ustream chat. Sri Harsha from Uppsala University says, I didn't quite understand as to how the modified cholesterol is triggering Parkinson's disease. Dr. Testerman, perhaps you could explain the mechanism as you see it?
Well, I wish I had my neuroscience colleague here to answer that, but cholesterol in the brain is very, very important, and it's very important to have the right levels, and there is some signaling involved with cholesterol. So we believe that this modified cholesterol is altering membrane properties, perhaps permeability or some other properties, and we don't have the exact mechanism worked out yet. We're going to that'll probably take several years to figure that out, but we do believe that it's not allowing the neurons to function normally, and over time it becomes toxic enough that the neurons die.
Hi, I'm Tina Say with Science News Magazine, and I have a couple of questions. First of all, are there any epidemiological studies in people to suggest that Helicobacter pylori and Parkinson's disease go hand in hand? And secondly, you found this effect in these mutants that didn't bind very well, and you said that they don't also infect very well. So what do you see in the wild type situation where the bacteria can bind well to the stomach?
Okay, great question on the epidemiology. Actually, there is a bit of history. Back in the 1960s, before Helicobacter was discovered, doctors noticed, and there was a publication on this, that Parkinson's patients were much more likely to have had ulcers previously than non-Parkinson's patients. And so at that time, ulcers were thought to be due to stress or diet or some other things. But once Helicobacter was discovered, some people did start looking at prevalence of Helicobacter in Parkinson's patients, and it was found to be more prevalent in Parkinson's disease patients. So there is a link epidemiologically. And as far as the different strains, the wild type does seem to also cause disease, but more slowly. So we want to look at some later time points so we can get a better view of what happens with different strains as you wait. I think it may be just that this mutant strain is working a little faster to cause the disease, and the wild type will take longer. But there are also some known virulence factors in Helicobacter that can influence inflammation, they influence cancer, and there is some evidence that one of them, CAG-A, may be associated with Parkinson's disease. So we want to try some bacteria that have and don't have this virulence factor to look at what role that might play.
We have another question from the audience. Several.
John Rosenblatt, Mayo Clinic. It wasn't quite clear that in your mouse model where the mutant that are infected with the mutant strain of H. pylori, have you kind of connected the dots in the sense that they're infected with the mutant strain? You can demonstrate the altered cholesterol. They also have lower levels of neurotransmitters such as serotonin, and they also have the altered locomotion pattern that you described?
Yes. We don't have a specific test for the modified cholesterol. There are some groups that study it that are able to detect it, but we haven't gotten that assay working in our lab. But yes, we do check the mice at the end of the experiment to make sure that they're still infected. And we look in, we do locomotor beginning of the experiment and then at points during the experiment and right before the mice are euthanized. So we do follow that, and then we look in the two brain regions, and we look at tyrosine hydroxylase, which is responsible for synthesizing dopamine. We see that coming down, and then also the dopamine levels as well. So it does seem to have all of the features of human Parkinson's and also Parkinson's induced by certain neurotoxins that are injected or given.
And another question from our audience here.
I'm Stanley Malloy. I'm from San Diego State University. In addition to the positive association with gastric cancer, there's this argument that Helicobacter has a negative association with esophageal cancer.
Right.
And that is something that I think carriage of Helicobacter as an adult is really important. Could you comment on that relative to how you view this elimination of Helicobacter as solving the problem?
The data that I've seen on the reflux disease have been a little bit conflicting. So there have definitely been studies that have shown that, yes, having Helicobacter infection decreases the reflux and the development of Barrett's esophagus, which leads to cancer. But there's some other studies that haven't found the link. So I guess it comes to a risk-benefit. I'm not sure that the possible benefit in reflux disease is worth the risk, especially since there are other ways of treating reflux to prevent esophageal damage.
All right, well, we seem to have run out of questions, is that right? And I think we can say that we're going to call this our first ASM Live session to a close. I want to thank Traci Testerman for speaking with us about her research, and thank you all for giving us some good questions. We have 2 more live sessions later today. I invite you all to return to our great fun, and we've got some more coming up in the next couple of days. Thanks very much. Again, I'm Jeffrey Fox with Microbe, ASM's monthly magazine, and that's it for our first session. Thank you.
Thank you for having me.
Yeah.